Morning.
Hey.
Quite a few people already. I'm going to start a quick poll while we wait for a few more people. If you guys wouldn't mind filling out these few questions for us, that'd be great. This makes me feel like I'm teaching a lecture again, having these polls.
Additionally, we are recording today's session so that we can send it out after. Make sure that you have a link to be able to rewatch anything that we go through. We'll go through a number of demos today and answer questions that come up. So hopefully this will be a helpful resource for you as you start doing your own double and triple label analysis.
Yeah, I'll be emailing out some useful links as well as the recording after the session. Okay. So quite a few people actually haven't tried Pipsqueak yet. John, I don't know if you're seeing the poll results.
Yeah, that's great. Well, welcome to all of those of you who haven't.
Oh, this is really interesting. Great. Okay. I'm going to go ahead and leave the poll up for just another 30 seconds or so, and then we'll get started.
Okay. For some reason the audio is not able to get it.
All right. Well,
okay.
Why don't we dive into it, Christy, and we can.
Sounds good. All right. Well, I will start. John's going to be giving the presentation today, but I will introduce us to start. So I'm Christy Lawton. I'm the Chief Science Officer at Rewire Neuro. And John Harkness is the CEO, and the two of us were actually postdocs together at Washington State University a few years ago. And now we're working hard full-time to bring you this software that hopefully will be useful for your research. And as I said before, I'll be emailing you guys after this to provide you with some useful links as well as the recording. We will stop a few times throughout this presentation to check for questions. Feel free to put questions in the chat. We'll be having a longer Q&A at the very end. So if you want to save the more detailed questions for that, you can always unmute your mic to ask questions verbally if you'd prefer, but please wait until the actual Q&A times for that. But again, feel free to put other questions in the chat. I'll be monitoring that. And with that, I'm going to pass this off to John, who will give you a lovely demo. And thank you all for coming.
Yeah, definitely. Thank you all for being here. It's really great to see so many users and potentially new users here. Kind of the general Zoom hygiene that we're all pretty used to at this point. If you're just watching for now, please mute yourself. Feel free to turn on your camera if you'd like, so that I can see someone else there other than myself. But also, feel free to interrupt me if you're really curious about something, or if I say something that confuses you. I'd rather answer your question now than have it burn until the very end. So this is fairly informal. Really, we just want to kind of show you some of the cool features that Pipsqueak is capable of, and walk through some of the confusing spots that I think pop up for some users. So today, I'm going to split the time into two different focuses. The first, just kind of giving you a little bit of background on Pipsqueak, and then generally how to get going with it, how to get it installed on your computer. But I'm not going to spend a whole lot of time there. I will answer questions after that, though, if anything pops up. But then what I'd really like to talk more about today is how to do co-localization analysis with Pipsqueak, specifically looking at triple label analysis. And you'll see why we're going to focus on that. But then we have a very exciting thing to announce or launch today. in that we've just recently this morning uploaded a new or opened a new version of Pipsqueak that enables you to start training your own models and adapt our models to be capable of what you're interested in analyzing. So we'll take a quick look at that and see how it works. So generally, Pipsqueak is a program that I first started developing about 5 years ago when I was a postdoc at WSU. And the reason for developing this was that in my lab we had a lot of undergraduates and we had some high school students coming in, and we wanted to share this microscopy and now this histological analysis process with all these people in the lab. But we had a problem of low inter-rater reproducibility. So when I would analyze my confocal images and I would ask an undergrad to analyze the same confocal images, we would hardly ever agree on how many cells were in that image or the pixel intensity of those cells in that image. And so I wanted to develop something that could start to standardize how we were analyzing these images. And really that started out very basic. We were just opening image J and kind of running a picture through it. Since then, we've actually published our early results and showed that the Pipsqueak can significantly improve the accuracy of analysis, particularly between two different raters. And that's I think a really important part of what it's capable of doing. However, one of the problems with this early version of Pipsqueak was that it was easily confused by high background, overlapping cells, kind of the typical suspects that you have for automating image analysis. And the reason for that was because early on, we just used really simple techniques like image thresholding that enabled us to kind of get an idea of where a picture was or where a cell was in a micrograph. And so, here's an example of these overlapping cells that would all be counted as one cell, or we'd have faintly or dimly stained cells that wouldn't get picked up. And then, probably most frustrating is when you have some, fiber track going through the middle of your image that would kind of disrupt that intensity or thresholding intensity. And so since then, we've actually taken this project on as a full-time gig, and I'm working with a really fantastic group of scientists and machine learning engineers to apply some of the machine learning techniques that are now available to us to actually see constituents within an image. So we use a lot of object detection and image segmentation that enable us to see a cell with a computer the way that the researcher is seeing the cell. And so these advances have really focused on kind of, I think, what the critical problem is with image analysis right now, and that's that there is truly a reproducibility crisis in biomedical research. There's a number of papers that have come out fairly recently, these two from Nature and JAMA, that are essentially saying that a large portion of research is not reproducible. And I think one of the reasons that we see that, obviously this isn't the entire reason, but one of the reasons for that is that we have a number of these assays that allow bias or subjectivity to creep in. And so, here's a really easy spot for us to remove some of the, human subjectivity for detecting cells or running an image through analysis. And so we've addressed that with our software called Pipsqueak AI. And so, this software is really designed to help you select your cells in a micrograph that's coming from a confocal microscope or from a fluorescence microscope, and then to help you determine the co-localization of constituents in that image. So our tools are designed to be helpful to the user rather than to replace the user. And I think that's a really important differentiation with some of the AI software packages that are out there now, is that we're taking a very, I think we would like to call an ethical approach to the use of AI for this type of thing, because I think it's easy to understand that relying entirely on AI to make these decisions for us can be very dangerous. The types of images that you're analyzing may not be what the AI is trained for, or the AI might just go down the wrong road. And so the way that we've designed Pipsqueak is to allow the human to be the decision maker that's benefiting from some automation tools that are developed or provided by AI. And I'll really show you what that means here in a second. Oops. But generally, I want to give you an idea of some of the work that we've put into testing the accuracy of our system. And these are actually some data that were recently presented at our SFN poster, this SFN virtual this year. But one of the things that we've spent a lot of time on is determining our inter and intra-rater reliability. And the way that we do that is essentially by testing our AI against a number of trained human raters. And so, kind of the summary of this here is that across a number of our early detection models, specifically we're looking here at WFA, parvalbumin, CFOS, and ADoxO-DG. I'll spare you the details on what these stains are if they're not important to you, but what we found is that our AI is very good at detecting what a human will detect, and where it really benefits the analysis process is that it's more consistent at doing that. So in terms of inter-rater reliability, obviously, if Christy and I both use Pipsqueak, we're going to be producing results that are very similar. But the other part of this that I think is really compelling is that each time I use Pipsqueak, I am producing results that are very similar. So this intra-rater reliability component of our analysis, I think, is very important.
Oh, John, it seems like maybe some people can't hear you very well.
Oh yeah, shoot, let's see. Well, let me know if I need to speak up anymore. I'll try to talk directly at my microphone. So right before we dive into kind of what Pipsqueak can do, I just want to kind of address some of the ways that people are using it. A lot of our users are focused in image analysis that are images coming from optical microscopy, but we're also looking at a whole number of targets ranging from fertility analysis to C. elegans and to cancer drug detection. And so to get a better idea of some of the places where Pipsqueak is being used, you can actually navigate back over to rewireneuro.com. And about halfway down the page, we have this great link that will take you back to all of the publications that have cited our 2016 paper. And this is, just kind of a quick list of where Pipsqueak is being used, but it's pretty neat to see it kind of branch out from some of my postdoctoral research looking at these extracellular matrix structures, and now expanding into a whole variety of places where perineural nets are important, such as spinal cord regeneration and obesity. And now we're branching out into new fields such as activated microglia and CFOS and everything else. So it's really neat to see where the software is now being applied. And so with that, let's just take a quick look at how to get going with Pipsqueak. The first place you're going to want to look is to head over to our website, rewireneuro.com, where you can download Pipsqueak by pulling up a demo license or a basic license, and essentially you can also just download the software here from this downloads page. And so once you've done that and registered a license, you've probably seen an email like this come through from Rewire Neuro that's giving you an order number and a license number. And so those are great because we're going to use those to get registered. But before we do that, if you head over to Get Started from rewireneuro.com, you can take a look at some of the installation videos that we have here for both Mac and Windows. So if you haven't yet installed Pipsqueak onto your Fiji or ImageJ software, then take a look at these videos here. So once you do have Fiji installed, or once you do have Pipsqueak installed, let's go over to Pipsqueak, and we'll open Fiji. So there's my Fiji bar, and I'll see Pipsqueak up here on my menu. I'm going to open it, and the first thing I'm going to need to do is to register my account. And so I can do that really easily by typing in my email address, coming up with a password, and then I'm going to take my order number here, copy that into order number, and my license number, copy that into license number. And the only reason that I'm really bringing this up is to say that you need to register once you get into Pipsqueak, even though you've already kind of gone through that process on the website. So great, once we've registered an account, I'm going to go ahead and log in. And now that I'm logged in, I can start to see some of my features, some of the things that we can do. The other thing that I'm going to show you here really quickly before moving on is if I've actually got a couple licenses or maybe a couple accounts, one of the things that I can do is go over to a brand new website we put together at my.pipsqueak.ai that allows you to log in and manage some of your licenses. So maybe you've gone through a basic license, you've tried it out, and then you've purchased a full license, so you can go in here, see which one you're using, maybe change to a different license and manage everything on your account. So great, taking a look at that. Now let's go back to Pipsqueak. Once we've registered and logged in, then we're ready to go. So let's pause here real fast. If there are any questions about how to get going with Pipsqueak, this would be a really good time to address those, or if you've had any problems as you tried to get going with Pipsqueak. And if not, then we can push right on. Oh yeah.
Someone has a question?
Yeah, great. So Olivia, I see, what's the difference between basic and AI? That's a really good question. And I think Jamie, I saw you maybe raise your hand. If you want to, feel free to turn on your mic here in a second, or you can just type it into the chat. But yeah, let's take a look at this real fast. So, you know, there are these two different versions. And one of the things that I think makes, you know, what we're doing different with Pipsqueak is that we're really sensitive to the role that this software has in research workflows. And so we're committed to always having Pipsqueak Basic available to users for free. Essentially what Pipsqueak Basic does is opens up your images, walks you through the analysis process. It can do single label, double label, triple label analysis with co-localization. But to detect the cells, it uses some of those basic ImageJ-Fiji tools such as image thresholding. And so this works really well in a lot of images. Where it doesn't work quite as well is when you have, you know, high background or unusual types of cells. Some of those perineural nets that I was looking at in my research were really the rationale for moving beyond this. And so what we developed was the AI capability that's available in Pipsqueak AI. We've set this up to try to be, you know, a really easy to use, affordable software that is something that you can actually share across your lab. You know, the software is something that you can log into with that user account. So if Christy and I both want to log in with maybe our lab account, we can share that. It just kind of works like your Netflix password, and that only one of us can be using it at a time. But yeah, that's a really good question. And Olivia, let me know if you want any more detail on that. And so, yeah, Pete, maybe that answered your question there a little bit too. So we're able to share these accounts. You know, again, we're really a research focused group here. And so, you know, we've got a number of ways to make this accessible for anybody. And we're willing to, you know, to put together a whole package of licenses or seats for a lab that would ensure that everybody in the lab is able to get on and use Pipsqueak at the same time. So, you know, with that, I think it's, you know, something that Christy and I would be happy to chat with you one-on-one about. Oops. So great, let's head back to the demos here real fast. And if there are any other questions that pop up, feel free to shout out. So in our last webinar, we went through some of the single label analysis, and I'll come back to this at the very end. But let's go ahead and focus in on co-localization analysis, because I think this is where you know, the benefits of using software like Pipsqueak really shine. That's because, you know, once we start to get into these co-localizations that include, you know, 3 labels or even 2 labels with a whole lot of cells in the image, it's very, very difficult to figure out where those cells overlap. And so, okay, what I've done here is I've selected triple label analysis, and the first option that I'm facing is essentially if I would like to analyze an image or if I would like to sum a Z stack of images. one of the ways that Pipsqueak has been designed is to allow you to import a stack of Z stacks that can be used for analysis. And one of the ways that we think that that is best done is by summing those stacks into a single 2D plane rather than trying to analyze a max projection image. So that's something that we've gone into in the last webinar, and we'll focus on again in the next webinar, but I'm not going to spend too much time on summing here. So let's go ahead and analyze this image. And what I'm able to do now with Pipsqueak, and this is a bit of a difference maybe from some of the versions that our users have seen in the past, is I'm able to select all the images that I want to throw into this. And so we're doing a triple label analysis. Rather than having to name our images with some particular convention, I'm gonna open my folder here that has the images I would like to analyze, and I can even just drag and drop them in here. So I'm gonna drag in the first image I want to analyze. Here's the second image I want to analyze, the third image here, and then, oh, in this case, I actually have a merged image, and that's going to be helpful for me to be able to look at the co-localization. And so I'm going to drop that in there. However, if I don't have a merged image, maybe I didn't export it from my confocal, I can leave that off, and Pipsqueak is going to work just fine without it. So I've got my images in there. I can also stop here real fast and select what stains I know these particular channels are. So in this case, I think my first channel is parvalbumin. My second channel is this WFA, which is a stain for perineural nets. And my third channel, I think in this case, is CFOS. So let's take a look at how that goes. Once I select them, Pipsqueak's gonna open them up real fast, make sure everything's there. And it gives me an opportunity to enter a little bit of information. This isn't something that's required for your analysis, but it sometimes makes that post Pipsqueak analysis a little bit easier. Once I get to Excel or to my statistical software, I might want to know, which experiment this is. So, oops. So this is going to be experiment 1. This is going to be, all right, subject 42. Let's go with that one. I like that. And, grouping control, this might be my treatment group. We'll just put it in something like that. So great, I've got that information. And now I've got a little confirmation here that these are the images I'm going to open. That looks great. If it's not, I could select no, but let's move forward. And finally, we're starting to see the analysis process start. So here I've got my first image. This is a parvalbumin image. And something else I want to highlight here, since this is new since our last webinar, is we're giving our users the ability to select just a particular part of their micrograph that they want to analyze. And this is, I think, important if you're, maybe looking at an area of brain tissue that has a lot of fiber tracts running through it, or even if you just have a very small nuclei, within your image that you want to focus in on. Limiting the analysis to a subregion is a great way to do that. So I've checked this box. I'm going to press OK. And now Pipsqueak is giving me a chance to just, draw the area that I want to analyze. And so here, I'm basically going to take the whole image, but I'll limit it down here just a little bit so we can kind of see how that works. So, okay, I've got that. I'm going to add that area to my ROI manager. Great, got it. And now I'm going to press OK. And so we're going to go back, and we are going to subtract out the background in this image that I don't want to focus on. One of the ways that I can do that is through an automatic background subtraction. It's just placing ROIs around the background, around the edge of the image, and subtracting that out based on a certain range of the pixel intensities that show up. If you want to learn more about that, there's a great description on our FAQ section on the website. In this case, I'm going to select some manual regions. And so here I can do that just by maybe drawing on the image here. So that's going to say background, that's background, and I think that's background there, maybe one more up here. Okay, great. So those are my 4 background ROIs. Press OK. And we're moving on. And so one of the first things that's popping up here is my AI prompt. And so what I'm doing here is I'm choosing the model that I want to use to analyze this image. And so in this case, it's the parvalbumin model. I can also choose some parameters of how that model is run, including the amount of overlap that I'm willing to accept of the ROIs that are returned, and how sensitive I want my AI to be. And this really goes back to the ethical AI that I brought up in the beginning, because what we're doing here is we're allowing you to drive the AI to help find cells. Our goal is to find the cells in this image, but I don't want it to do all the thinking for you. And sometimes I want it to be a little bit more liberal in how it finds the cells. And sometimes I might want it to be a little bit more conservative in what it describes as a cell. And so I can change that right up here in my detection sensitivity. And I'll show you that a little bit more detailed in a second. So I'm going to press OK. And great, what it's doing here is it's uploading the image to the AI server. It's processing it. It's already got it. Wow, that's super fast. And that looks really good to me. So I've got two images here. Basically, I have my raw image that doesn't have the background subtracted. And then I have the image that I've subtracted the background out of that we're actually going to measure the pixel intensities from. But this looks really good to me. I think that definitely captured all the parvalbumin cells here. And that's a great indication of how well that AI model is working. So I'm going to say, okay, I like those ROIs, and I'm going to go forward. I'll describe some of those other settings here in a second, but we've definitely talked about that in more detail in our first webinar. So all right, I've got my WFA image here. This is my second one. Let's go through this real quickly. I'm just going to accept the automatic background subtraction here. You can kind of see how that looks. Cool. And I'm back to my AI window. So here I'm going to keep kind of the same sensitivity settings, and I'm going to say okay. See how it does here. So these are kind of interesting structures, because it's actually an extracellular matrix around the cell body, and so they're fairly hard to analyze, and they're fairly hard to describe to an undergrad, as I experienced as a postdoc. It's really difficult to agree on exactly what is a cell here. And so I'm gonna blow this up just a little bit so you can kind of see. There we go. You can kind of see that it did a really good job. The AI did a really good job of both detecting those cells, even some of the dim ones, but it also did a really good job of detecting where some of these cells overlap. And so, that's what I like to see here, like between these two, is those are very close perineural nets, but we've differentiated two. And so, I like this pretty well. Actually, here's maybe an example of a cell that I'm not so sure about. And so what Pipsqueak does is it gives me an opportunity to say, nope, I actually don't like that one, and I'm going to delete it from analysis. And that's really where this ethical AI comes into play, is I'm using this tool to help decide, or to help speed up my cell detection, but it's not doing all the thinking for me. So great, let's accept that. We can say our ROIs are complete. And finally, we're moving on to CFOS. And what I want to show you here is just how fast this AI works when we are looking at these really busy images. CFOS is one of those that I always hated doing by hand. Same thing for 8-oxo-DG. It's an oxidative stress marker that we often used in my postdoc lab. And so here I'm going to turn up my sensitivity just a little bit, because I really want to detect all these all these CFOS stained cells, and I'm going to turn down my overlap just a little bit, because I want to keep everything there for the most part. And so what that's doing is it's essentially telling the AI that I want to see the most liberal definition of what is a cell here, and that looks really good to me. Here again, I'm going to zoom in just real quickly so you can kind of see, that we've detected just about everything. Oops, I missed one here on the edge, maybe just because that's that's on the edge of our image. But this is essentially taking an agreement of what we've trained to be a CFOS cell previously. And so, for my lab, that works pretty well. However, that might not necessarily be what you want to see in your CFOS model. And so we'll come back to that in just a sec. I'll show you how we can alter that. But in this case, we're just doing triple label analysis. So I'm going to go ahead and push forward here. And what we're going to see is Pipsqueak is actually going through now, and it's taking all these ROI sets that we've generated for those 3 cells. And it's done it so fast, it's beat me to the punch here. But it's gone through and it's identified everywhere where we have a double label occurrence of, in this case, WFA and parvalbumin. And so this is a really important step in our process, because it's saying, okay, of all these ROIs that you've shown me, these are the, I don't know, 7 or 10, whatever, that are co-localized, but I have a chance to, again, select if I agree with this or not. And so, this is maybe a place where I would say, I don't think that that's the parv cell that was on the WFA cell. And, in this case, I think it all looks good. So I'm going to accept it. But if need be, I can go in and tweak that selection. So here's a more difficult one. Now we're seeing all those CFOS cells line up with all the WFA cells. And so there's a lot of CFOS and there's a lot of close together and big WFA. So we'll see how we do there. And voila, wow, that looks really good to me again. I think I really like where all that CFOS is lining up. But here, let's take an example. So if maybe I don't think that that's the right CFOS cell there, I have a chance to go in here and just nudge it around a little bit so I can select the CFOS cell that I think is the culprit. Say I want to change that one and move on. And so once I do that, it brings it back up and I can choose to move forward, or I can choose to move the WFA. And so lastly, we've got our third triple labels, or our third double label set, which is CFOS and PARV. So great, that looks good. Let's move forward and we'll take a look at the really complicated part of this is now it's taking all of our double label sets and looking at how those overlap for our triple label analysis. I think this is where co-localization can become almost impossible to do by hand. I really don't know how I would go through and do all of that CFOS staining to for these 3 cells or three stain types. And so I think that looks really good. But here's a great example of maybe where I disagree with it. In this case, this is one big WFA cell that has some overlap with both CFOS and parvalbumin. And so this would be a chance where I might just delete that whole thing because I don't think that's actually triple labeled. In this case, let's just move forward so we can take a look at something else. But that is essentially how we can get to triple label analysis. And when we do that, Pipsqueak does a really good job of spitting out some results that are easily imported into Excel. And one of the things that I think is really efficient about this is I have all of my different double and triple and single label populations separated out into this log window. And so, I can simply select this entire window, copy it, and take this over into Excel and import that, straight into Excel columns. And I'll show you a little bit more about that here in a second. But this is a great way to just kind of speed up going from from image analysis to data analysis. So various morphologies. Microglia, that is a fantastic question. And actually, let's go ahead and just move straight on over to that. I was going to show you another triple label analysis, but one of the things that I really want to highlight here, because we just launched this morning, is our ability to adapt our model capabilities. And so let's do that by taking a look at microglia, because we don't have a model to look at microglia yet, but we can. And one of the ways that we can do that is by either going into single label or multi-label analysis, going to analyze, and we're going to open the image that we're looking for. And so in this case, I have a microglia image right here, because this is one of the ones that I wanted to talk about. And so I'm going to open it up and I'm going to press run analysis. And boom, there's this big, beautiful microglia image. And so, there's, okay, I can put in my metadata there if I'd like, and I'm going to kind of skip over it. But there is, kind of a weird focus thing going on in this image. This isn't my image. And so I'm going to limit my analysis here just a little bit. So in this case, I'm going to just kind of draw into the center of the image. I'm going to cut out the edge of the tissue, something like that, let's say. Okay, great. And now I'm going to open that image up for analysis. So, again, here we're gonna do our background subtraction. I'm just gonna kind of breeze through this real quickly here. So let's take a background ROI over here, and a background ROI over here, and I don't know, a background ROI over here. Cool. And so finally, what I might be trying to do here, if I was going through this image on my own, is I might be just looking for an easy solution. And so let's go ahead and try the PARV model. We've really got it tuned up here. It's going to be very sensitive. let's see if parv is able to pull anything out. It's certainly not what this parv model is trained for, but we can see how it does. And oh, okay, actually, I mean, it did it did pretty good there. That's kind of interesting to see. And then we have some really low confidence ROIs that popped up here from the AI. So those are probably ones that I would want to remove out or just reduce the sensitivity of my AI. And so it's going through and just changing these ROIs into squares. That's one of the things that I asked it to do in the settings window. And we're not gonna have time to get to that settings window today, but this is something that you can easily adjust for your own.
We'll do that as another webinar though.
That is another, that's right, yeah. Okay, so that model's okay, but I wanna go in and I wanna adapt this to be my own. And so for my model selection drop down, I'm going to go to create a detection model. This is brand new. So you probably haven't seen this before. And I'm going to say rerun. And so now when I do that, it's popping up this kind of long-winded window here that's giving you an opportunity to name this new model that you're going to develop. And so in this case, this is microglia. So I'm going to go with John's have a microglia model. When I'm choosing this name, and the thing that I want to point out here is that it's important for me to keep that consistent across all the images that I want to include in this training set. And essentially what you're going to do is you're going to put together a set of images that can be used to train, that model that's specific for your marker, whether maybe it's a better parv model for you or it's a microglia model. But by giving it a consistent name, we're going to know that those are the images that you want to include in there. And the other thing that's important about this, and you'll see here in a second, is when I go through and find the cells that I want to include in this parv model, it's really important that I find all of the cells that I want to include in this parv model. So I'm now training the computer to look for whatever it is that I want to identify. And what that means is that if I put in a cell here that is not what I want to consider a microglia, an activated microglia, then I'm going to be giving the computer bad information. And if I include here, maybe an example, if I include, a cell like this, an ROI like this that's cutting my cell in half, that's a bad example for the AI. And so I really want to be kind of conscious here of what do I want to end up in the end, and am I spending the time to get that right? And so just a little bit of editing here, and rather than going through and editing all of these cells, let's just imagine that we have. And one of the really fast ways to do that, I think, is by taking one of your ROIs, and you can use the T key to stamp it around on all the cells that you want to include. So I don't know how that's showing up over Zoom here, but you can stamp that, and that's essentially a fast way to put those ROIs around everything. But great, let's accept this. So I've got it. There are maybe all the microglia that I want to include, and what it's done now is it's sent that up to your own file on the Pipsqueak servers. What we're doing now with this kind of beta release of our adaptive AI modeling is giving you the opportunity to put as much into it as you want, as many images as you want into that, and then just asking you to let us know that you're done. So if you send Christy or I an email, or just send an email to our general email account, then we can now take that little set that you've put together and adapt a model to be specific for you, and then release that model to you so you have it available. But while this is in beta, there's just kind of that one last manual step to it. So great, that was all that we had to do there. I've got the beginning of my new model trained up, and we're ready to go. So all right, what other questions do we have here? Can this software, sorry, Christy?
I was just wondering if you had already addressed the question about calcium imaging.
That's a really good question. And so, yeah, that is something that we are working towards. We are able to analyze video, and we are able to identify or analyze images like calcium imaging. But one of the ways that we're going to do that, I think, most likely is through a partner in this area. And it's not something that we've fully put together yet, but it's coming. And so I can tell you to keep an ear out for it. It's something that we're working towards hopefully very soon. And so, all right, so for someone who has 400 plus microglia images to analyze, oh, thank you very much, Olivia. So to create different models for each microglia activated state, 3 key ones that you're interested in. Yeah, I think one of the ways that we've found modeling to be most effective is to either look at those different markers that you're looking for, different cell types in different channels, or to to really focus in on the specific types of the cell that you're interested in. So if you're looking for 3 different states of the microglia, I think one of the ways that we'd want to really focus in on that training is to look at a number of examples of each one. And so that might be a good chance for you and I to talk about how to train that model, Olivia, so that we can make sure that we're accurately identifying each one of those 3 states. But that's absolutely something that we can do, and is a great use of Pipsqueak. And so if you have any other questions, feel free to write them in there. I'm maybe going to go back and let's take a look at one other thing that we can do here, because I think this is an interesting or an important tool in Pipsqueak. So have you tried software to analyze mRNA signal rather than cell type? That's a really good question. We haven't, but that would be another. So this is a question about analyzing mRNA signals. And so that would be something I think we'd really like to work with you on. So again, maybe just a question to take offline, and let's continue that over email and see if we can come up with a way to do that for you. Oh yeah, and great question there on how many example cells are required for training. So in this early beta package, we've found that you can get down to as few as 10 images, for some sort of transfers. One of the places that we've done that is specifically transferring over from our parvalbumin model to detecting CFOS. These are both kind of round cells that are plentiful. And so, that was a fairly easy transfer to do. And we were able to do that with just about 10 images of, around 10 cells on each one. Some of the transfers are going to be a little bit more difficult. And so right now we're expecting to, potentially go up to 30 images. But as we progress from our beta implementation of this adaptive modeling to the full release, it's called the non-beta, what we're going to be implementing are some new segmentation models that should help you with that, annotation generation. And so this will be something that will become easier and easier over time. And so yeah, 10 images rather than 10 cells. And so, that can end up being, 10 to 100 cells per image. And so, really the idea here is the more examples that you can give the model that you're trying to train, the more effective it's going to be. But, our entire focus right now is developing this into a process that's as painless and easy as possible to create a really accurate model. So yeah, let's take a look back here at triple label analysis, because one of the things that I think is really interesting is that sometimes we might go through analysis several times, and one of the things that happens when we do that is that we might just want to tweak 1 or 2 of our markers, but not have to redo an entire image. And this kind of goes back to our ethical AI thing. So all right, let's take a parvalbumin image. Let's take another WFA image, because that's what I've got here in front of me. And in this case, let's take one of our 8-oxo images. And I'm not going to give it a merged image. Oh, well, okay, let's give it a merged image because it's pretty. And so I'm gonna say okay. And so as we go through this, we're gonna again have the chance to enter information, do our background subtraction. I'm gonna kind of breeze through that, and let's get over here to analysis. And so great, I've got parvalbumin queued up. Let's go through parv. Voila, oops, I've got my sensitivity super high again, so I'm just gonna bump that down. Kind of this mid-range I think is really good for a lot of our models, because generally we train them to be fairly conservative in what they describe as a cell. So that looks really good to me. Awesome. I'm gonna move forward. And I've got the WFA image here. So again, I'm just gonna subtract out the background real fast. Boom, WFA, AI analysis. Going through, that looks like there's some really faint staining here that is kind of interesting. These images are just really cool to look at. All right, so I'm just going to accept that for the sake of brevity here. And finally, let's go to our 8-oxo image. And one of the things that you'll see here is that, I think there are like between 300 and 500 cells in this image. So you can imagine the amount of work that went into in postdoc analyzing these things by hand. This was definitely one of the reasons that why I wanted to develop Pipsqueak. Doing these by hand is terrible. And so the AI is able to do this really quickly. That looks really good. Oh, great. I'm gonna bump up my sensitivity here just a little bit. I want to see if I can get, some of those really faint 8-oxo cells. And we'll see how it does. That looks a lot better, but maybe I've actually already done this once before. So I'm redoing my analysis. So this is the tool that I want to show you. We're able to go back into this load previous ROI set. And I think this is fantastic if you want to, change the background subtraction that you've done, or maybe you want to just subsection part of an image. And so I'm able to go in and look back to, the analysis that I had previously done. And in this case, let's see, I think I have my old ROI set. Well, I have my old ROI set hiding from me, but that's okay. So we're just going to take an old ROI set here from another. Oh, there we go. So here's my old ROI set that I've already done for this image. And so I'm just going to say open that. And what it's done is it's just replaced the annotations, the ROIs that were put on most recently with the ones that I had developed previously. And so, this is a really nice way to go through analysis quickly if you want to start over without having to redo the entire thing. Because I know that a lot of work can go into developing an ROI set. And so great, let's kind of put a pin in that and go back to the questions here. And so yeah, Olivia, I see your question here. Are we able to do bulk analysis? Yes, we are, and that's one of the really interesting features that is in development right now. And so, currently we've got this set up so that you can specify the exact files that you're looking at pulling into Pipsqueak, and really direct analysis at that. But we are able to do bulk analysis, and so again, this is kind of a feature that's requiring just a little bit of input from you on how you want that to run. And so you'll see more bulk analysis come up here in the future. Right now, it's really best to go through and do these one by one, but we're gonna make it possible for you to drag in a whole folder of images and run through them, especially with things like single label analysis, where you might just want to run through a whole bunch of maybe CFOS images all at once. Those are pretty easy to do. And so yeah, Matthew, your question here, train it to do 20x images. Yeah. Extend the customized parameters to tiled scan. Really good question, Matt, and thanks for bringing that up. One of the things that I hadn't shown here, so I'm going to cancel out of this image, is that yeah, we are absolutely able to to train for a different focus of image coming in. And one of the things that we've put a lot of work into with Pipsqueak early on was having our models general enough that we were able to move from like a 20x to a 40x image and still have accurate cell detections come up through that. And so we've been able to do that okay. If there's a slight tweak on that would be benefited from some additional training, that's absolutely something that we can do. But one of the really neat things that we can do here is to take a tiled image and actually break it up into something that would be possible, something that we could break up into small enough objects that we could analyze it here. So this is again not my image, but one that a collaborator is giving to us. And wow, you can see it's enormous. It's way bigger than my screen. And so we're gonna take the whole image, and I'll zoom out here in a second so you can see it. There we go. Cool. So this is a bunch of tiled images for perineural nets put together here. And so if I go through this with my WFA stain, I think those cells are actually probably a little bit too small for us to detect, even with fairly high sensitivity settings on here. And so let's just give it a go. Let's see what it does, even though this image is huge. Okay, yeah, so we didn't get anything there. That's okay. But now let's go in, let's change our settings to actually break this tiled image up into some smaller parts. And I'm going to do that by telling this that I want to make a 5x5 grid across the image. And let's try WFA again. Let's see if this works now. Oops. Say rerun. Okay, great. So I'm rerunning the image here. And oops. Well, all right. I think I chose a bad image here to show this off with, but that's the way demos go. So you know, essentially what's really great about this is it does make it possible for us to analyze some really, really large images and make the whole thing possible. Although you just have to feed it the right settings sometimes. So anyway, Matt, I'm happy to go into more detail with that, and Olivia too. It looks like I'm happy to go into more detail about our image sub-segmentation, either offline, or we did go through some of that in our first webinar, which I think is available on our website. And if not, maybe we can email out that link again.
We'll be redoing that one soon as well. And I will be emailing out a recording of this, just addressing Michael's question there for that.
Jamie, great question about DAPI. That's actually a model that we have in development internally right now. I think you should be seeing that one here fairly soon. You're right, DAPI does show up pretty well with you know, some of the other models that we've got available. I think CFOS is a good one to start with. But one of the really cool things that we can do with DAPI, and this is kind of an upcoming feature that I don't have here to show you right now, is we're able to use DAPI as kind of a way to seed some of our detection. So, you know, if we think back to like those 8-oxo images that I showed you, where, you know, we've just got these cells all over the image that are fairly diffuse and hard to identify. When we first started doing that analysis, we actually used DAPI as a way to find the nucleus of the cell and then expand our ROIs out from there. This is just a really kind of clever technique to, you know, hone in on where we might need to be looking. But yeah, that's one of the ways that we will use DAPI, but you'll also see that DAPI model come out here pretty soon. Great, and then can Pipsqueak run a stack of TIFF images generated in Fiji? So right now, we're really focusing on 2D analysis. So, you know, summing those stacks together is generally the best way to pull out all the information, unless you wanted to look at each 2D plane, each Z step individually, in which case, yeah, you'd be able to, you know, run through each one of those and potentially even use those as co-localizations if you wanted to see, you know, how your cells are developing down through your Z stack. Another feature that we have coming out here in the near term is the ability to import your LIF or CZI file so that you can take those images straight in from your microscope rather than having to, you know, export them and sum them or however, you know, you're bringing them into Pipsqueak. But yeah, really good question about Z stacks. Let's see. Yep, so we will have a recording of this coming out soon. Probably send that out by the end of the day. So well, thanks for coming. And 3D images, that's a really good question. And that's one that we've talked a lot about internally. And actually, I think I've got a little example of that we put together. So 3D is something that we've focused on, and we've actually developed some models that are capable of looking at cells in 3D space. Again, this kind of goes back to the Z-stack question. And, you know, really where we're at right now is trying to determine how useful this is going to be to our users. So let's see, Varus, that would be something that I'd love to talk with you more about offline, because, you know, we've done a little bit of 3D analysis in our own labs. I know this is something that Christy has done at times, but you know, finding really good applications for that technology is you know, kind of where we're interested in exploring more. So yeah, let's continue talking about that. So Kelsey, if I have WFA measurements from a triple label output, which measure represents intensity of each cell? Really good question, and thanks for reminding me, because I kind of glossed over our results. So let's take a quick look at here at some of the results that came out of our analysis. So let's see here. We've got the CFOS analysis that we did, and so one of the things that I've got coming out of CFOS is my log print. So this is everything that showed up here in my window, and I think it's really useful to again copy paste this straight into Excel. But sometimes I want to be more specific about, just the particular population I'm looking at, or I might want to build our results files out from just specific cells. And so in this case, I can go into each one of my image folders that are resulting from analysis. I can see the single PARV, single WFA, single CFOS. I can see all my double label stains, and I can see my triple label stains. And so here, maybe let's look at our double label results, and what you'll see. Let's look at this. Let's look at our single label, and what you'll see here is this comma separated value file. And so this is really fantastic because I can double click that, import it straight into Excel, and when it opens, you'll see all of the CFOS images here, all the CFOS cells here that were in that image and identified. And so I've got a whole lot of different columns up here. And what you might find is that some of these columns are more important to you than others. And so, I'll tell you, in our research, we often did not spend much time looking at, the area of the cells that we were looking at, or even just this mean value. Where we would spend more time looking at the mean minus background value. And so, essentially this number here is often what the SORG lab reports in terms of measuring the intensities of the perineural nets in our research. And that number is just calculated by subtracting, the background from our mean value. Some other labs that are using Pipsqueak are more integrated interested in the integrated density of the cell. And so that's a really interesting calculation that is essentially taking our mean minus background and multiplying it by the area of the cell, and it gives you a little bit more an idea of how that cell structure or volume is changing with the intensity of the stain. I'm not going to dive too much into which one you should be using or the science behind any of these outputs, other than to say that Pipsqueak is really adaptive, and we're able to integrate any of the metrics that you're interested in having fairly easily. And so as we begin to develop more flexibility in what's printed out, we're able to just hard code that in right now for you. So if you see something, or if you have something that you're not seeing here, reach out to me and let me know, because we can definitely come up with another metric for you. Additionally, we've got a lot of information here about the particular cell that's being analyzed, and that's fantastic because you can now carry these identifying numbers across the different ROIs in your analysis to say that, oh, okay, this ROI here 005 00001 is the same cell that I saw in the other channels. And so that's really important. So Kelsey, I hope that answered all of your question, and if not, let's continue talking offline.
A few more questions.
Yeah, so we're just up here against the hour, and I want to be respectful of everyone's time. But Christy, if you've got an eye on this, maybe hone me in on one question here.
There's just a few more, I think. So 2D images does that include max projections? Yeah, we usually work with those.
Yep, absolutely. Any image that you import. So if you decide that you'd rather look at max projections rather than summed images, it's going to look at them all the same. If you are able to import the TIFF or JPEG or PNG file into image J, we're going to be able to open it in Pipsqueak. So it really doesn't matter if it's max projection or summed. That was just a little bit more based on how we were exporting images from our confocal in my lab. And so, yeah, Olivia, the difference between the log window and results window on my screens, is essentially that we're getting just a little bit more information in the results window. We're able to get, this mean minus background, or I'm sorry, we're able to get this integrated density. We're getting, these ROI numbers, but a lot of that information is carried through. And so this is just a little bit of insight on where we're going with Pipsqueak, is essentially to provide, more and more ability to sort your data. So where we would like to get is for you to, indicate which columns you're interested in seeing, and even which groups you'd like to see an average or a mean from, and being able to export your data just like that. So you're going to see a lot of advancements coming through pretty quickly here in how the data are reported to you. But for now, I think if you find the metrics that you're interested in here in the results files, that's where you should be focusing. Otherwise, the all results folder or all results file is a great place to start. And so Varus, yeah, that's right. So you did understand correctly that we can create new models optimized for your stain by just uploading several of those images with your own analysis on them. Really the important part of that is just indicating what it is that you're looking for by putting those ROIs onto the image. And so I think it's a really great way to just adapt some of these models to be specific for the particular stains that you're looking for, and letting that AI be as helpful of a tool as possible for you. And so, yeah, Olivia, in terms of when we'll release the LIF files or the ability to import LIFs, all I can say right now is that it's coming. We're currently really focused on our adaptive modeling, and so once we start to roll out more and more advanced versions of that, we're going to shift over to some of these other tools. But that's really good to hear that that's maybe important for you. And so yeah, keep your eyes peeled for it, because it will definitely be on the horizon.
So John, it's just past the hour, so we should probably wrap up. I'm going to go ahead and show a quick post-webinar survey, but you guys asked some great questions. Thanks so much.
Yeah, so while you're taking a look at that post survey, we'd love to hear what you think of all this, and maybe what would be useful to go through in the future. I'll just point you back to our website here real fast. As I said earlier, Pipsqueak Basic is free. We're actually giving out a 2-week demo, so twice as long as normal, but a 2-week demo of the AI tools if that's interesting to you, since you came to the webinar. And so Christy's going to send out a link for that, or I think we actually already did send out a link for that yesterday. So check that out. There's really no downside to trying it out, seeing if it works for you. And if it doesn't, I think one of the things that makes the Rewire team special is that we're happy to see if we can make it work for you. We're really hands-on with this. This is our focus, and so let us know if there's a feature that you want, or let us know if there's a way that would make Pipsqueak more usable for you, because we're pretty excited about building a community of people who are using this software. It's definitely been important to my research, and I hope it can be helpful to yours. So thank you again for coming. Look forward to these emails coming out, and if you haven't checked out the website, check it out at rewireneuro.com. So thank you, everyone.